Hi everyone, and welcome back. Before we begin, I'd like to take a brief moment to introduce AM Academy. AM Academy was founded with a clear mission to connect knowledge and experience from the lab, the field, and the public health. We are strong believers of the concept of One Health, recognizing that meaningful progress can only be achieved when these three areas collaborate and inform one another. So over the past year, Animalia has also invested significantly in strengthening its research department. We have successfully established reliable and reproducible challenge models for PRRS, for glasser disease, and post-weaning diarrhea. Meanwhile, we are broadening our focus to include additional pathogens, continuously develop scientific expertise and tools required to advance in vaccine research, and also to assess innovative pharmaceutical solutions. So stay tuned for the upcoming AM Academy episodes where we will dive deeper into these initiatives and share further insights from our ongoing research efforts. So today's guest is María Montoya, an immunologist with a long, fascinating scientific journey. She began her career with a master's degree in immunology at the University of Birmingham, followed by a PhD at the Universidad Autónoma de Madrid. Then she moved into a postdoctoral and fellow scientist roles at the Edward Jenner Institute, where she deepened her expertise in viral immunology and immune responses to infection. Over the past decade, Maria has become a leading figure in pig immunology, studying how the immune system responds to viral infections and playing a key role in the development of innovative vaccine strategies. She has led research groups at both CRESA and the Pierre Bright Institute, and she is currently the group leader of viral immunology, therapies and vaccines at the Consejo Superior de Investigaciones Científicas in Madrid. Maria's relationship with Animalia may be recent, but we are truly delighted to have contacted her. So in this episode, Maria will focus on the well-known old virus influenza. She will walk us through the latest advances in research, especially from the immunology perspective. She will also explain how this growing knowledge can be useful for future research and its impact in the field. So welcome, Maria.
Thank you very much, Ariadna. It's a pleasure to be here and to be the second one in this Animalia series of seminars. And it's really challenging, and I'm very excited about it. Thank you very much.
Thank you. So before we get started, I would like to let you know that at the end of the session, we will have some time to answer some questions. So to ask a question, just click on the button next to the microphone. Below, you can click this button at any time and it will turn color purple, but just wait until the end of the session where we will give you permission to open your camera and microphone and ask the question, okay? So let's get into some of the questions I prepared. Maria, could you please share what are the main advances of your team and what are the main focus of your research right now?
Well, thank you very much for this question. It's very interesting that you make this question because, as you said, my basic knowledge is immunology. But in the field of veterinary, my understanding is that you need immunology in order to design either therapies or vaccines. So what we have done in all these years that I started to work in pig immunology was understanding pig immunology and antiviral immunity. So in this way, we have designed and we can design new vaccine or improve vaccines that are already there, as well as therapy. We are focusing now, and we have done a lot of work in innate immune responses because these are the first ones that are mounted. So if you can control that, everything that comes later will be directed by the first innate immune responses. So we have done a lot of work in the dendritic cells, in interferon signaling, in the way they control major pathogens like influenza, PRRS, or African swine fever virus. So we have done a lot of work in cytokines, in controlling, these kind of immune responses, controlling and controlling immune responses, which sometimes induce pathogenicity, which is important to understand. So at the moment, we are dissecting innate immune responses in influenza viruses. That's why we did an experiment there in Animalia, and we had the chance of being there and enjoy the beautiful place that you are settled in. But we are very much focused in the first stages of infection, because we think that if we can modify that, whatever comes later will be influenced by this, mounting these early immune responses. So if we can change that by any ways of therapy or vaccines or any kind of, thing that we can do to strengthen the immune responses, then I think the animals will be stronger to respond to a viral infection.
Yeah, you're right. I mean, in the end, influenza is like a very acute virus, right? Very quick. So that makes sense. Another question, you had the chance to work at several renowned institutions alongside with different experts. How has that variety of experiences influenced the way you approach immunology research?
Well, that's a very interesting question, because I think that we think that whatever we do research is the most important thing in the world, and we are focusing to that. But when you change environment, change countries, change continents, because I have worked in, you mentioned that Oxford, but I have worked in Argentina, in Australia, and you see different approaches, and you see the different diseases that are in other parts of the world, but most importantly, how people face this kind of challenge. And you learn from that, and you learn about different tools that people are using. You have the chance of discussing, different approaches, and I think it's very rich in order to have a broader knowledge about a question. So I always recommend everybody to do some kind of a stage, or in whatever they can, to move from different environments, because it gives you flexibility in your thinking that is very useful to approach complex diseases like the one affecting pigs.
Yeah, of course. Now, getting deeper into the main topic of our talk, which is influenza. So understanding that influenza is remarkably a fast virus in animals, and that it doesn't usually circulate through the bloodstream, it is only logic to believe that the innate immune system is critical during the infection. So what key questions about the innate immune system in pigs do you think still need to be answered to improve the understanding of the influenza pathogenesis?
Well, influenza, as you said, is very much focused into the respiratory tissue, and it's not systemic as others, but we see systemic effects. Sometimes we see a little bit of temperature, we see some cytokines in serum. So even though the virus replicates in the respiratory tract, you see some effects all over the body of the animal. Yeah. I'm talking about pigs. And also when the animal is us as well, we have that. So, because the virus sometimes is most of the time is the same. So we have to these are two different approaches. So where the virus replicates, which is the respiratory tract, the effects of the replication could be in the respiratory tract, but it could also be systemic. So we have to differentiate that. So, but we need, as I said, we need to understand two main things for me in the influenza infection. One thing is the mucosal side. Mucosal, in terms of immunology, in terms of delivery, in terms of vaccine, is a world that is not that explored, and we need to focus into that, because for respiratory pathogens, they enter through mucosal tissues, and this is the first thing that they encounter. This is where they replicate, the first cycle of replication. So if we want to vaccinate, for example, we need to have IgA in the mucosal tissue. I mean, the fact of having IgG in blood doesn't help to protect the animal. So we need to have IgA. We need to have the particular cells in the mucosal tissue. I mean, if you have a lot of T lymphocytes in the spleen or the lymph nodes, they don't know whether the virus is starting, entering or not. So we need to focus into mucosal tissues in terms of delivery, in terms of replication, in terms of immune responses. That is very important. And secondly, I think, as I mentioned before, how innate cell subsets in pig, particularly distinct macrophages, distinct dendritic cells population, coordinate antiviral defenses in the first stages of infection. So this is important, and also whether these first stages could lead to immunopathology, because sometimes we see immunopathology because this is an uncontrolled immune response that could damage lungs or other tissues. So in terms of influenza, this could be a problem. And I think we need to understand, to answer these questions and to improve our understanding, what is the replication and the immune system in the first stages, and what is the impact later in the infection. And we need to know that. We know that the sequence of influenza is important. We know that some proteins are important in terms of blocking innate immune responses, like NS1, for example, in the case of influenza, but there are some others that also play a role. So we need to understand more that in order to, design strategies to counteract these effects. So.
Yeah, right. So the innate immune response is like really, really important for the control of influenza. But we are always like currently developing vaccines and studying like which is the best approach to control the disease in more like a vaccine type of way. So the intramuscular inactivated vaccines are known to induce antibodies responses, but also fail to trigger this mucosal immunity that you were just talking about. How do you think this affects the ability of these vaccines to be able to protect the pigs in the end?
Well, if you have IgG in the serum, you don't have any protection in mucosal tissues. So therefore, the virus is entering, is replicating. So the protection of the pigs will be starting when the virus will reach the areas where IgG or other cells or other arms of the immune responses are there. But at the beginning, what we want is to have, protection at the very early stage. We would like the pig not to be infected. Let's put it this way. Right. So the vaccines that we have now, they don't prevent infection. So the virus is replicating to a certain point where they find T cells, IgGs, B cells secreting antibodies and all of that. But at the beginning, there will be, 2-3 days or 2-3 rounds of infections where the virus is replicating. Whether this affects the health of the animal, this depends on the, sanitary status, the farm, and everything. But the thing is that we don't have vaccines at the moment preventing the entry of the virus. We prevent the disease, let's put it this way, the disease.
Right.
It doesn't prevent that the virus infect the animal.
Like in a very general conclusion, you would say that current vaccines act too late in the disease.
Well, I would say there's room for improvement.
Yeah.
So for example, something that could be delivered intranasally would be much better. Something that it would be delivered easy and in mucosal tissues would be an improvement. Something stimulating mucosal IgA would be an improvement. Something in the case of influenza, something that is fast. Taking into account that the virus fast mutates, something that is fast, that we can change the sequence very fast, following what the virus is doing, could be an improvement. So there are, as I said, there's room for improvement.
I see, I understand. But there's also another issue regarding the vaccines, and it is that sometimes it has been described that vaccine-associated enhanced respiratory disease happens in challenge pigs and that have already been immunized. So this is something that we would not like to see in our pigs that we vaccinate. It's contradictory. Do you think this is like an issue, like a major issue in the development of vaccines against influenza? And why do you think it happens?
Well, as I said, there's another point of improvement. Because these are generated by antibodies that are non-neutralizing. So you generate a lot of antibodies that are non-neutralizing, and therefore they bind to the virus, but the virus goes on and generates an immunopathology, basically. And nowadays, as I said, we don't need to generate these antibodies. I mean, these were done when inactivated vaccines were used and so on, as you mentioned. But I think nowadays, one of the things that I devoted a lot of years and other groups as well is you don't need to use the whole virus. You may use part of the virus, antigens. So which antigens need to be introduced in a vaccine to be effective? And then you remove the rest that are giving you this kind of side effects. So this is something that, I think we need to be more specific, but at the same time, we need to take into account the mutations that the virus is introducing all the time. But for example, nowadays with mRNA vaccines, for example, that could be an option, that you introduce the antigens that are needed for neutralization, but not the other ones. And then you don't have the whole virus. And therefore, my bet would be that there will be less side effects, particularly for non-neutralizing antibodies that induce this kind of viral defect.
Right. So despite the influenza research having advanced a lot, I know you're an expert in immunology, but generally speaking, what do you see as the main challenges in influenza research?
Influenza in pigs.
Or maybe if you have an answer for other species.
No, I mean, because it's different. I mean, we have protected mice from influenza 1,000 times, and it doesn't work neither in human nor in pigs. So, I mean, we have saved the world in the mice 1,000 times, but it doesn't work for the rest, So mice is a nice model, but it doesn't work in real life. So that's what I say in pigs, because in pigs you have to take into account different things. And also talking about influenza, you have, poultry, birds, and the problem that we currently have with H5N1. So these are different things to have to take into account. So I think we need to, at the moment, we are following the virus. We see, okay, there's a mutation here. Okay, we introduce it in the vaccine. Well, there's something here, and then we introduce it. I think nowadays with new technologies, as I said, with antigen definition as well, and particularly with new, I don't like the name, but artificial intelligence, which I think is not intelligent, but using IA, I think I have already seen a couple of papers trying to get, not going behind the virus, but going in front of the virus, trying to guess possible mutations of the virus, try to guess, possible antigens that could be introduced in a vaccine. I think we are at the moment in a very interesting cross point in where we can maybe do some suggestions of where the virus is going, and therefore whether we can be there before the virus arrives. You know what I mean?
I understand.
This is very interesting, and particularly with poultry or with pigs, which the populations are not as broad, because I'm talking about farm animals, not wild animals. Wild animals is a different story, but for farm animals, I think if we do a serious studies on that, maybe we can have something ready, off the shelf when the virus strikes, and then we can protect our animals.
Are you positive that these kind of predictions will be very useful in the future?
Maybe not in the very near future, because this is very new. But I would bet, we have been following the virus all our lives, talking about influenza. So the vaccine, particularly for humans and for pigs as well, and we are using already viruses and sequences that are not longer there or have been mutated already. So we are always behind. So somehow these kind of tools, prediction tools or models, may help us in the future to go in front of the virus. That's how I see it. I think that would be very interesting.
And these models have only been prepared for influenza, or also other viruses such as PRRS, for instance, that also continuously mutates?
I mean, I'm following influenza, I have to say,
Okay.
but I mean, I would say that could be, yeah, that models could be done also in other species. But as I said, mRNAs vaccines are vaccines that are, I mean, at the moment, vaccines are done either in tissue culture or in eggs.
Yeah.
I mean, and this takes a lot of time. mRNA vaccines, you can make them very easily and very, very fast. That's the most important thing. And even though we cannot predict how the virus is mutating, we can nowadays use mRNA vaccines faster than if we use tissue culture or eggs, for example. This is an improvement. And then, we need to guess maybe where the virus is going. But in terms of influenza, we have another layer of complexity, which is the jump from different species. I mean, we have seen, that we had the first pandemic of the 21st century, which was influenza A, and that was coming from birds, pigs, and then to humans, and going, jumping from different species. So this is something that we cannot predict at the moment, although there are some predictions. But the last clade that is currently infecting, particularly in Spain, had some mutations from the prediction, but some others that are not. So the virus is already surprising us. All the viruses are surprising us. So anyway, this is to make a long story short. I think we are starting to have tools that could give us some advantage from the previous approaches.
Yeah, step by step. So you already mentioned that the mice model works perfectly, but then when you translate this into pigs or maybe also birds, it doesn't work as well. From a lab perspective, would you recommend to always use like species-specific models, challenge models for each species, or do you think we can generalize a little bit?
I mean, nowadays as well, there are more. Sorry, I didn't mention that, but there are other models that not only mice, but also there are organoids. There are organ on chip that are starting to be used, that are modeling what happens in an animal, in your target animal, but in the lab, and these are becoming very, very useful because you don't need, as I said, we have been curing cancer, in mice all over. We have been curing all the kind of diseases in mice, and then, when it comes to the target species, it doesn't work. But it gives you some basis to start doing research. But now there are some models that are in the lab, like ex vivo models that can be used, like organoids or some organ that for the target species, and they are already for pigs, for cows, for cattle, for some other species that can be used. And I think these are much more informative than curing any disease in mice.
Right. Okay. Interesting. Another question. So maternal-derived antibodies play a pretty important role in the protection of young piglets. How do MDAs influence the piglets in terms of influenza?
Well, they are very good. They are very good for the piglet. So in fact, when we do our experiments, we need to, for the maternal antibody to wind down in order to infect the piglets for an experimental infection. So this is very important. So if we could get the B cells from the mother and transfer it to the piglets, that would be perfect, but it doesn't work. So maternal antibodies are very, very good, and they help a lot. And there has been some approaches to induce, maternal antibodies in mothers to protect the piglets. In influenza, they work very well, and for some other disease, there have been attempts to do so. But the thing is that when the antibodies come down, then the piglet, this is the window where the piglet start getting all the infections after the weaning. So we need to think how we can keep these kind of antibodies in higher levels. So, yeah.
Right. So another question, maybe a little bit a different topic, but I also wanted to mention that in Animalia, we studied the gut microbiota in terms of first infection, how it changes, how this virus influenced the protection in the gut of these pigs. And I know you also had some papers published about the lung microbiota when pigs are infected with influenza. Could you please develop a little bit on that?
Yeah, I mean, I have some slides on that. Because
Yeah.
I mean, we are very interested in the lung-gut axis. So we have started to do this work in collaboration with Dr. Stanis Nistal. And if you show some slides that I sent you, yeah, see that? Yeah, if you make full presentation, please.
Isn't it already?
No. Now okay. I can see you again.
Sorry, let me.
No, you're right. It's all right.
Okay, I think now. Is it better now?
That's it. That's it. So if you click again, next slide. So we have analyzed, influenza immune responses in different stages, but we wanted to have a look at whether, the microbiota that is in the lungs already changes the immune responses. So if you go to the next slide. So what we did was collecting lungs from different areas of Spain, from infected pigs. These are in different farms, in different places, and with different, all positive to influenza, of course. And then we did a sequence and analyzed the minion sequence and analyzed the microbiota that had, by 16S RNA sequences that they were in this mice. This was published September last year, in case you want to know more about it. Next slide, please. So I'm going to just touch into the surface of the information from this paper, but basically what you see here is that in the infected animals compared to healthy counterparts, we have more pathogens, more microbiota in terms of numbers. If you go to the next slide, then when you analyze which are they, you can compare each column and see from different samples what are the classes, order, family, and genus from different microbiota, and you see the difference already in this graph. Mm-hmm. If you click next slide, please, and then you say from different classification of bacteria, when they are infected or not, whether they have this bacteria or not. Next slide, please. So, in terms of relative bacterial abundance in the lungs of the influenza virus infected and non-infected pigs, you can see that there are some that are in common. That was something that we were expecting. And in the healthy ones, we had some others that are only in healthy animals, but we had more than 600 bacterial genus that were particularly detected only in influenza-infected lungs. Next slide, please. So, as I said, I'm not going to go into details. You have the reference on the paper there. And what we have seen in these animals is that not only is the quantity, but also the quality of microbiota in the lungs of these animals that is very different. And we have seen some genus classification that are more abundant in infected compared to healthy pigs. So we need to dissect that. But the next stage, the next study well, these are the people. Sorry, these are the people. Next slide, please. Yeah, these are the people, and as I said, this is a collaboration with the group of Stanis Nistal in CEU and my group. So we are, now that we know what happens in the lung, we want to analyze the axis of lung-gut microbiota, and because as I said, the virus is replicated in the respiratory tissue, but we suspect that there could be influences in the gut microbiota. So we would like to do this kind of studies, and we are doing that right now. So as I said, it's not that the virus is just in one tissue. It's that the consequences of that infection may be reflecting some of the changes along the body of the animal that doesn't necessarily have to have the virus itself, but it's a consequence of the virus in other parts of the body.
Right. I think these results also coincide a little bit to what we observed with the PRRS infection. We focus on the gut, but we also saw like quantity was higher in pigs infected with PRRS, and the quality was also lower, or there were more bacteria involved in inflammatory pathways.
I mean, I suggest you have a look at the lungs, because maybe, there's a connection, because PRRS replicates in the lungs as well.
That's right.
You have the virus in both places, whereas influenza is only in the lungs. So I suggest you do both, because maybe you find something interesting.
Yeah, you're right. Now let's change a little bit the topic. I want to focus on the recently outbreak here in Spain. I know it was avian influenza, but from your perspective as an immunologist and also influenza expert, what makes this outbreak particularly concerning? And what does the situation tell us about how well prepared we are when we manage emerging diseases and viruses and threats in animal populations?
Well, I think I need to answer you with, One Health, global health. We need to understand more about that, and to take into account, that virus jump from one to another without, distinguishing, who we are. But anyway, yeah, very sad. H1N1 outbreaks in Spain, affecting not only farms but also wild birds. This is very serious and very worrying. Mm-hmm. But I think we are not prepared. We are not. We need to. As I said, be polite. There's room for improvement. There's room for improvement, because anyway, from a immunological and veterinary perspective, this kind of outbreaks underscore our gaps of knowledge. Because when they come, we are not ready for a vaccination, we are not ready for any treatment, and we slaughter the animals, or they die massively, and this is very, very sad. So as I said, fast evolving vaccines like mRNA could be an issue, but the question is, what do we put in the vaccine? I mean, now we have mRNA, right? I mean, just to get the example of coronavirus, of SARS-CoV-2, we knew that if we include the S protein, it was protective. That was a major advance. But in terms of H5N1, what do we include in the mRNA vaccine? So what antigens, what epitopes needs to be included? So this is research that needs to be done. But I think we need to be ready for H5N1, H7, and whatever. So viruses that are very extremely dangerous and pathogenic for our birds and us and our animals. And we should have some kind of preparation of the shelf to stop it spreading, as it has been happening in Spain in recent months. So I think we need to have something, kind of. I don't know whether the CEPI as coalition of vaccines, and I have been speaking with people from the UK to do something similar in animal health, to have something prepared off the shelf, that when you have an outbreak, you have something quickly to give your animals, your farms, and stop spreading. We need something in terms of One Health approach. We need to have something. But it's very difficult when you ask a company or a government agency, okay, give me money to prepare something for a disease that is not already here. But I think this is the way to do it. I mean, we think that these are the risks, these are the pathogens that are hitting or could be hitting our industry, our animals very hard. Mm-hmm. And I think we need to be prepared before it comes. So, but that is asking for money and research before this before this pathogen is here. But I think that's the way to do it, because when it comes, if you stop the first outbreak, with something, with a vaccine, with a treatment, then you protect the rest of the farms and the rest of the
right.
So you need to do it, let's say, in days. And in days, you cannot prepare a vaccine, you cannot prepare a treatment.
It has to already be prepared.
To be preventive rather than, treating the disease. And for that, I think we need to change our minds in terms of being prepared. And being prepared means money to generate vaccines or treatment before it is here. And
right.
That still we are working on that.
But you were talking about a preventive treatment.
Preventive treatment or preventive vaccines that you will not use. You will keep it, in the fridge.
I understand.
In case it comes. I mean, this is what CEPI, this coalition for preparedness of pathogens in human, is doing. And I think we need something similar for animals. And actually, that is something that I suggested in a meeting in September last year in London, a meeting in animal health, suggesting, okay, we need something similar. And actually, I call it, I made a name for it called Chappy. I think we need something to be prepared for diseases that we know if they come here,
it's going to be devastating.
For the animals, for industry, for all of us, and and it may, in the case of influenza, it may have consequences for human as well. But there are some others like ASF, as we know, what has happened already in Catalonia. They are devastating for pig industry, for our animals, for wild animals as well, for wild boars. So we need to be prepared for that before it comes. That's my message.
Yeah, and you are right. Thank you for telling us your opinion on that. Right, so understanding that influenza evolves like very quickly, do you believe in autogenous vaccines in terms of being a possibility to manage an influenza kind of outbreaks in the farm?
Well, autogenous vaccines were used, for its purpose at certain time, but I think they are not as safe as they should. And I think we have some other tools at the moment that can, as VLPs, mRNAs, recombinant viruses, and some other tools that give us more, they are safer than autologous vaccines. I mean, I think that they were useful for a certain period of time and certain diseases, but I think we have other tools nowadays that they don't depend on. I mean, autologous vaccines are not a standardized solution by themselves. They depend on robust diagnostics, sequencing capacity. I think we have all the tools now.
So you would not recommend them in an endemic?
Well, I mean, depending on the situation, depending on the pathogen, depending on the things. I mean, there are some other, but I mean, there are, in animal health, there are pathogens that are, from the research point of view, very interesting, but very terrible, very devastating. Yeah. So if your only solution is autologous vaccine, this is better than nothing. But, and also it depends on the area of the world. As I said, you work in different areas of the world. Some other places, I mean, chances of having new developments of vaccines or treatment are very, 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 very little. So if your chances of protecting the animals is autologous vaccines, if this is your chance, use it. It's better than nothing. So it depends on the vaccine, depends on the area of the world, and depends on the pathogen.
Right. Well, thank you. Right, Maria, this talk has been wonderful. So thank you for your time. If there is one key message that you would like the listeners to take away from today's discussion, what would it be?
What would it be? I think, we need to collaborate more, industry and research people doing not only immunology, but other areas of research. Because sometimes people come to me and say, okay, we developed that and it didn't work. And we don't understand why. If it works, nobody wonder why, you know.
Yeah.
Everything is fine. But if it doesn't work, then people are going to say, why didn't you ask me first? And then we could have, you know, done it in another way. So I think a collaboration between companies and research in animal health should be encouraged. And we are here to help, and we are here to collaborate. I mean, all this initiative of public, what is in Spanish is colaboración público-privada, collaboration and private-public collaboration, and all this kind of proposal. I think we need to. In England, for example, this collaboration is much enhanced, and it's very fluent. Whereas in Spain, still there are some, you know, upside down, and you know, I think we need to, because we have a common objective, which is you know, the health and the welfare of our animals. I mean, this is the most important thing. So if we are there to help. So that is my message, basically.
So when you're talking about industry, you're talking about pharmaceutical companies as well as the swine
industry.
Industries, right?
Yeah. I mean, I'm talking about everybody who's involved in any animal management, worrying about animal health. I mean, this is it's important, because if you want to understand what is happening and why this is happening, you need some kind of expert. But you know, as a collaborator, as a, you know, advisor, as a, you know, project, as a research, whatever. But I think we need to talk more, because we have a common aim.
Right. Well, thank you very much. And before we wrap up, we would like to hear from the audience if there are any questions. Now is the time to share them. I think there's someone that has a question. Maybe, Christine, you can give this person permission to talk. Okay, so this person is not here anymore, but I have a question from another person that was unable to attend today. So I'm going to ask this question on behalf of this person. So he asked me, in an endemic farm where there's some outbreaks, small outbreaks of influenza from time to time, when would you recommend vaccinating those animals, or would you prefer not to vaccinate them?
Well, that is an interesting question, because it depends from the point of view that you look at it. If you look at it from the point of view of the gain of the animals, and you know, how healthy or how you know, they have, you know, that's one point. And another point is like, okay, I want healthy animals, even though they will not gain much weight. You know what I mean? So it's different. But I will always say to vaccinate if the vaccine matches with the strain that is circulating.
Okay.
So I would, because sometimes people vaccinate with the strains depending. I'm not going to go into commercial, you know.
Yeah, I understand.
But sometimes the strain that is used in the vaccine doesn't match with the strain that is in the, you know, circulating, and they say, okay, we spend that much money and it doesn't work, and it doesn't work because the same as happens in humans, the virus has changed, and therefore you are not protected, or your protection is, you know, 30-40%, you know, very small. So you need to know what is circulating in your farm, and then adjust the vaccine, whatever is in the market, to what you have in the farm. Because sorry.
Correct me if I'm wrong, but is it possible that vaccines, the strains they use on the vaccines are not well updated nowadays?
As I said, this is something that companies, this is a message from companies generating vaccines.
To update the strains,
but because if it is not updated and it is very different, you know, the vaccine strain is very different, your percentage of protection is going to be low. So you need to know, first of all, what is circulating in your farm, and secondly, you know, whether you have a vaccine that helps, because otherwise you're going to spend a lot of money and you're gonna disturb your animals by vaccinating with something that will give very, very little protection.
Right. And I have another question. In order to control the influenza like outbreaks inside the farm, like the endemic outbreaks that happens, do you think personnel that works there are like a key issue, and maybe we could take some like actions to prevent like from worsening, especially in winter and stuff?
Well, yes. I mean, as I said, the virus doesn't mind if you walk in 2 legs or in 4 legs. I mean, they basically. So it is us infecting our animals, and it's our animals infecting us. And if we go from one barn to another, we may be transmitting taking into account that is respiratory transmission. So in terms of influenza, security and management is very important in order to prevent our animals to get any disease from us and the other way around. So we need to understand that and to use, some protection for the animals and for ourselves. I mean, masks and all kinds of protection, because we are the ones,
transmitting it. Yeah.
I mean, we are the ones going from one barn to another.
Yeah, of course. And also the ones that like then we spend time with other people that also brings like infects us with other diseases and stuff. So
yeah, I mean, if you go to the underground to the tube in Madrid, you're gonna get for sure. I mean, I said to people, I mean, why do you ask me to have a mask? I need a mask to go to the tube. I mean, in Madrid. I said Madrid, but it could be any other city. If you go to a place where there's a lot of people sneezing, coughing, blah blah blah, you're gonna get something. I mean, for sure. And then after that, you go to a farm, and then you got it. I mean, you do it with all your good intention, but you need to protect your animals from yourself and the other way around. You have to protect yourselves from the animals that they could have some kind of virus circulating, virus or pathogen.
Yeah, so maybe like something to like sum up this kind of 2 questions would be like, first, check your personnel, improve your like
security,
protective equipment and stuff. And then if you still have disease, uncontrolled disease, then maybe check the most appropriate vaccine and vaccinate the animals.
Yeah, check whatever is circulating and then check what is. But for example, for that, we can help. If a company say, okay, we have flu, but we don't know what it is or whatever, I mean, we can help. We can say, okay, you have this flu, and whether it match with vaccines. I mean, this is something that we can do, and there's not a lot of trouble for us to do that. And or we can say, okay, this is what you have in your farm, and there's no vaccine covering that. So don't spend more money, on that, because vaccine is not going to help. I mean, it's going to be very disturbing for the animals, and it's going to be a lot of money, and then it's not going to help you.
That's right.
So that's why I said we need to talk. We need to talk, and we need to, help each other, because that will be in the benefit of our animals.
Right. So, well, a heartfelt thank you, María Montoya, for joining us today.
I mean, I enjoy it a lot. I don't know whether there is I have here one person wants to talk. I don't know whether it is someone who...
yes, this person left the meeting just before I said that.
Okay, it's a pleasure speaking with you, Ari, and as I said, we are in CIB-CSIC in Madrid, but if we can be of help, we are always happy to collaborate and help with everybody. It has been a pleasure being here, honestly.
Yeah, thank you. We really enjoyed it as well. I really enjoyed it personally.
Thank you very much.
Yeah, looking forward. Our next session will be in March the 26th, and Enrique Vidal Barba from CRESA will join us and talk about the lumpy skin disease. Okay?
I hope I will be able to enjoy and to say hi to Enrique.
Okay. Oh, maybe there's someone that wants to talk. Alex, I see here. Alex, you need to give permission to the computer to open your microphone and camera as well. Okay, I don't hear him. Do you?
No, neither do I.
All right. Well, thank you very much, Maria.
A
See you.
pleasure.
Bye.
Bye-bye. Bye.